Hello everyone, good evening, good morning, depending on where you are. My name is Denise Doolan and I am the past president of the International Society of Vaccines. It is my great pleasure to welcome you to this August edition of the ISV webinar. So it is my great pleasure to introduce as a speaker for this webinar, Professor Eui-Cheol from Korea. So Professor Shin received his MD in 1996 and his PhD in 2001 from Yonsei University College of Medicine in Seoul, Republic of Korea. He then received his postdoctoral training from NIDDK at the National Institutes of Health in Bethesda, Maryland, USA. Professor Shin then joined the Graduate School of Medical Science and Engineering, the Korea Advanced Institute of Science and Technology in the Republic of Korea in 2007, where he is currently a professor. He also serves as the director of the Center for Viral Immunology at the Korea Virus Research Institute at the Institute for Basic Science in Daejeon, Korea, which he has served since 2021. His laboratory performs research on T-cell responses in human viral diseases and cancer. In particular, he is currently focused on T-cell mediated pathogenesis, senescence of T-cells, reinvigoration of exhausted T-cells, and immune response in SARS-CoV-2 infection. Professor Shin was elected a Fellow of the Korea Academy of Science and Technology in 2019 and a Fellow of the National Academy of Medicine of Korea in 2024. So it is our great pleasure to welcome such a distinguished vaccinologist to give their talk today. And his talk will be on the phenotypic and functional characteristics of SARS-CoV-2 vaccine-induced T-cells. And can I just ask that all panelists and participants, if you have a question, you have the option to either add it to the chat function or raise your hands at the end of the presentation. I'm sure the Professor Shin would be very happy to answer questions. So in the meantime, please join me in welcoming Professor Shin for this wonderful presentation. Thank you.
Yeah, thank you for kind introduction, Denise. And from now, I'm to share my slide and let me, thank you for giving me this opportunity to present my recent work. And today, I'm going to talk about the SARS-CoV-2 vaccine-induced T-cell response in terms of their phenotype and their function. So as you know very well, so after infection or after vaccination, so host, we can get a neutralizing antibody and memory T-cell response. In the case of neutralizing antibody, the antibody can protect the host from the infection itself. So we consider that this neutralizing antibody is one of the main axes of this vaccine-induced effector function. But also we need to think about the T-cell response, especially in terms of the variant. So as you know very well, the SARS-CoV-2 variant very easily escape from the neutralizing antibody response. So during the COVID-19 pandemic, we knew already all this kind of the escaping by variant, so continuously. I want to emphasize that this SARS-CoV-2 variant very hardly escape from the vaccine-induced memory T-cell response. So why? So there's the importance is about the distribution of this epitope location. In the case of neutralizing antibody, in the case of SARS-CoV-2, the neutralizing antibody target very narrow region of the spike protein. But in the case of the T-cell, the epitope is scattered across all the spike protein, and also in the case of T-cell response, the different individuals have different HLAs. It means that they have different epitope sequence. Due to this, the diverse HLA allotype, the T-cell epitope is not concentrated on some certain narrow region. So that's the reason why the SARS-CoV-2 variant cannot completely escape from the memory T-cell response. So at the beginning of Omicron, the emergence, so I remember that it was 2021, the late November. So my lab, we immediately tested this hypothesis. So it means that, in other words, so we want to test that the Omicron variant, how it escaped from the memory T-cell response elicited by this Wuhan-based mRNA vaccine. So in that time, we recruited individuals who got the vaccination 2 doses of vaccination of the Pfizer vaccine, mRNA vaccine, and then 3 doses of vaccination. And then we perform very simply is the intracellular cytokine staining by using ancestral sequence, spike OLP, the Wuhan-based sequence, and Omicron in that time, BA.1 based overlapping peptide sequence. So as you can see from this representative figure, the Wuhan OLP sequence spike can induce this interferon gamma response in this individual like 0.07%. But in the same individual, when we switch this the OLP with the Omicron sequence for the stimulation, it was 0.06. So it means that a little bit attenuated, but still substantial response could be detected. So now I'm showing you the data by this graph. Compared to the ancestral spike sequence specific T-cell response, this Omicron OLP, the specific T-cell response, was statistically significantly, it was reduced by 20% or 10% in terms of interferon gamma response. So there's some significant reduction by variant, but so still just 80%, in other terms, 80% or 90% of this memory T-cell response is still preserved. So in that way, in that paper, we conclude that this vaccine-induced memory T-cell substantially, considerably respond to the new variant, Omicron variant. But such kind of the data was published not only by our own group, but other group. So it was already well known that due to such kind of the many paper, so it's well regarded that the T-cell is hardly escaped by the new variant. So on the basis of such the publication, well, once John Wary, once in his review article, he summarized very nicely about the role of the neutralizing antibody and memory T-cell response in COVID-19 pandemic. For example, so we can imagine 3 different situations. Someone gets the vaccination, and then there's no variant. Then this neutralizing antibody will maintain their high neutralizing antibody titer to the virus. In that case, this individual, if he is exposed to the virus, then this neutralizing antibody can protect host from the infection. But it was not the case in the COVID-19 pandemic, because even after Omicron, continuously, even even until present time, the time to time, continuously, the new variant escaping neutralizing antibody emerged. So our current situation is more like the middle, the figure. So after vaccination or previous infection, many people have the neutralizing antibody, but against the previous strain, so not against a new variant. In this situation, we can imagine that this host have the low titer of neutralizing antibody. But still, if he get the previous infection or vaccination, he or she have high memory T-cell response. In this case, upon the exposure to virus, maybe due to the low titer neutralizing antibody, this host will be infected. But the T-cell response can protect this host from the severe disease. But without any vaccination or without any previous infection, so if someone have just the very low titer neutralizing antibody or low level of memory T-cell response, then maybe the host will get very easily the infection, and after infection, the severe disease. So will be progressed to the severe disease. So many people in the current era, we have such kind of the high memory T-cell response and low titer neutralizing antibody titer. So with this background, from now, I'm going to share the data with these 4 different topics. So in my talk, I'm going to touch each topic just a little bit about this, a very unique topic, very interesting topic. And from the first topic, I'm going to talk about the T-cell durability and generation of the stem cell-like memory T-cell. The stem cell-like memory T-cell, this story, it started even before vaccination. So in 2020, the first year of COVID-19 pandemic, we recruited these convalescent who have the previous infection. In that time, there's no vaccine, so they didn't get any vaccination. But so we recruited the convalescent who have the infection and after spontaneous recovery, we followed up the recovered patient for more than 10 months, like more than 300 days for their memory T cell response. And we evaluated this memory T cell response by ELISpot assay or intracellular cytokine staining or AIM assay by diverse T cell technique. And we found that during the 10 months, the follow-up period in this the recovered patient, recovered individual, the SARS-CoV-2 specific T cell memories were maintained. We showed that in this paper. But so at the same time, we want to know what's their differentiation status of the memory T cell. Especially, we want to focus the stem cell-like memory T cell. Even before COVID-19 pandemic, the virus infection, like a yellow fever vaccination situation, the stem cell-like memory T cell, this concept was well established. The stem cell-like memory T cell, in terms of their phenotype, in the CD45RA or TCR7, this the traditional, our definition of this in the FACS plot about the memory T cell. The stem cell-like memory T cell, they look like naive T cell, but they are not naive T cell because the real naive T cell don't express CD95, but the stem cell-like memory T cell, they have 95 positivity. In that way, we can distinguish the TSM cell from the true naive T cell. But then what's their function of the TSM cell? So in the previous paper, they showed that the TSM cell have a very high self-renewal capacity by IL-7 or IL-15. And also more importantly, they have multipotency to repopulate the broad spectrum of memory and effector T cell. It means that TSM cell, after their proliferation, they can differentiate to diverse subset, TCM or TEM or TEM9 that way. And also previous literature, they showed that TSM cells are required for the long-term protective T cell immunity. So with this background, we want to look at the TSM cell differentiation in the COVID-19 recovered individual. So now I'm showing you this AIM assay data. By AIM assay, activation-induced marker assay, we could detect the SARS-CoV-2 spike-specific T cell in recovered individual. And as you can see here, for example, in CD8 T cell, this cell population in terms of CD45RA and CCR7, they look like naive T cell. But almost all of them, naive-like T cell, they express CD95. It means that they are not true naive cell. They are stem cell-like memory T cell. And we examined the TSM cell, their proportion among SARS-CoV-2 CD4 T cell and CD8 T cell. We found that during the 10-month follow-up period, the TSM cell are well maintained. And also we confirmed the TSM generation by tetramer staining. And then for the CD8 T cell, we used the MH class 1 tetramer, and within tetramer positive cell, so like 4.9% of the tetramer positive cell looks like the naive T cell. But almost all of them, nearly 100% of the naive-like T cell, they are TSM cell in terms of CD95. In that way, we confirm again the generation of the TSM cell after natural infection and spontaneous recovery from the infection. In addition to the detection of the TSM cell, in our previous publication, we also showed that from the SARS-CoV-2 recovered individual, really the TSM cell have high probable capacity. In this case, we simulate the TSM cell with the spike protein. We confirm that they have really very high probable capacity. And also, we also confirm their multipotent differentiation ability. So in this way, in this case, we sorted TSM cell and stimulate them with the spike OLP. And the several days culture, we examined again their progeny cell. And we found that among progeny, not only TSM cell, but also, oh, sorry, this was some typo. Not only TSM cell, effector memory, central memory, or such kind of the diverse subset, memory subset could be detected after proliferation. And also, we confirmed their high the self-renewal capacity by IL-15 stimulation. In that way, not only the phenotype, but also in terms of function, we confirm their stem cell-like function of TSM cell, which are specific to SARS-CoV-2 spike. So in this, our previously the publication, we showed that the stem cell-like memory cells are nicely developed after spontaneous recovery. But so in our second, not only in this the infection situation, also after in our second study, we extended the TSM cell concept to our vaccine cohort. In the second year, 2021, from the beginning of 2021, also in South Korea, so many people got the vaccinations, COVID-19 vaccine. And we recruited the healthcare worker who got the Pfizer BioNTech, the mRNA vaccine, the 2-dose vaccination. And then we obtained the peripheral blood PBMC from the healthcare worker cohort. And now I'm showing you the MH class 1 tetramer staining, the data from one of the single individual as an example. So before vaccination, the tetramer positive cell are hardly detected. And after first vaccination, the tetramer positive cell was detected. And then second boosting, the huge increase of the tetramer positive cell. And during the 6-month follow-up period, the frequency was dramatically reduced. So not only the single individual, but also now I'm showing you the data from our cohort. So before vaccination, pre-vaccination, the baseline, and after first vaccinations increase, and then after second vaccination, more increase, and then 6-month follow-up period decrease. And by using the 6-month follow-up period, 6 months memory T cell population, we confirm again their memory, the function by proliferation assay, by after peptide simulation for this T cell epitope, and also interferon gamma production ability among tetramer positive cell. But then our next question was the T cell memory durability. So because we hope that the memory T cell maintain very well, but it was not the case. Especially some people have the significantly reduced response during the 6-month follow-up period. Then, so we also look at their differentiation status within the tetramer positive cell, effector memory, TEMRA, central memory, and also stem cell-like memory T cell. So within tetramer positive cell, the stem cell-like memory T cell are gradually increased during the follow-up period. And then we calculated CD8 T cell memory T cell response, their durability. In this case, we divided the MH class of multiple frequency at 6 months by the MH class of multiple frequency at the peak time point. In that way, from the previous slide, it means that we divide this value by this one. In that way, for each individual, we calculated how the CD8 T cell memory response durable in each individual. Then we want to know what determines the T cell durability, memory durability. Then, so we found that finally, the TSM cell generation very nicely correlate with the durability, especially TSM generation immediately after 1 or 2 weeks later than booster vaccination. This is the value, so nicely correlate with the 6 months durability. It means that in other terms, we can say that if someone develops TSM cell after second booster vaccination, then he or she will get the highly durable T cell response. So considering the function of the TSM cell, the very high proliferation potential and then cell renewal capacity, it is very understandable data. And also as a second question, we also ask this one. Then who will get more nice generation of TSM cell? And then simply we found that the aging of the host inversely correlate with the TSM generation. So we can say that young people have the better TSM generation and more longer durable CD8 memory T cell response. So this one is in this paper we show that the CD8 T cell, the durable T cell response, is nicely correlated with the TSM generation after vaccination in the case of mRNA vaccine. And then, so we extended the TSM cell, this concept in the further study. So now we consider that the TSM cell generation after vaccination will be good biomarker for the durable CD8 T cell response. Then, so this now I'm sharing with you one of the unpublished data. In our next study, we compare different vaccine platforms. So in this, another healthcare worker, the cohort, we recruited the mRNA vaccine, Pfizer-BioNTech mRNA vaccine, and also AstraZeneca adenoviral vector vaccine, the 20 and 20. And then especially, we compare the memory T cell response after second boosting vaccination. After second boosting vaccination, the mRNA vaccine and adenoviral vector vaccine, it boosted the T cell response after second booster vaccination. And it seems that the frequency of the CD8 T cell response, memory T cell response, looks a little bit higher, the better in mRNA, in the case of mRNA vaccine compared to AstraZeneca vector vaccine, but not so significant. But most striking difference was demonstrated in the analysis of the TSM differentiation. We found that within this the SARS-CoV-2 spike specific T cell, gated by the AIM assay, we analyzed the differentiation status of the SARS-CoV-2 specific CD8 T cell. And we found that the TSM cell, the better generated in the case of adenoviral vector vaccine compared to mRNA vector vaccine. And then, so we confirmed the TSM generation, not just by phenotype, also by function. In this case, we took the 6 months follow-up, the memory stage, 6 months the stage sample, and then we stimulate them, the PBMC, the CD8 T cell, with the spike OLP. We found that the mitotic index means that in the case of CTV assay, the mitosis, the antigen-specific proliferation, was significantly higher in the case of the adenoviral vector vaccine-induced memory CD8 T cell compared to mRNA vaccine-induced CD8 T cell. So, and also, I didn't bring that slide, but in the case of the heterologous vaccination, in that time, 2021, among some healthcare worker, so we could recruit a sample, the individual who have first vaccine with the AstraZeneca and second vaccine with the BioNTech, the Pfizer-BioNTech. In the case of heterologous vaccination, so the TSM generation and the mitotic index looks like they resemble more the AstraZeneca vector vaccine rather than the mRNA vector vaccine. So in this is my final slide in the stem cell-like memory T cell generation, this section. And so now we are doing ongoing study, then how, even in older people, how we can enhance the TSM cell differentiation after vaccination. So maybe we think that maybe we can use cytokine adjuvant in the case of this mRNA vaccine or AstraZeneca vector, the adenoviral vector vaccine. But anyway, this is our ongoing study. So in my second part, I'm going to show you the T cell response. So what will happen in T cell response after breakthrough infection? So we are going to show you the breakthrough infection induced T cell response in terms of in the aspect of a certain specific epitope. So in Korea, like other country, so many people have the breakthrough infection after 3-dose vaccination. And then 2022, the BA.1 the Omicron, the breakthrough infection. So we obtained the peripheral blood mononuclear cell from this cohort, 3-dose mRNA vaccination, and then after the BA.2 breakthrough infection. Then we compare the T cell response against the original spike, Wuhan-based spike, and BA.2 spike, the T cell response. And then after the breakthrough infection, we simply just compare the T cell response difference CD8 between the Wuhan spec T cell response and BA.2 spec T cell response. Especially in the case of CD8 T cell, we found that BA.2 spike spec T cell response is much higher than Wuhan spec T cell response. So this is very understandable because the individual, they already experienced BA.2 breakthrough infection. But then, so when we look at this T cell response in each epitope level, we found very interesting story. So now I'm showing you again. So in the previous slide, we just used the overlapping peptide as a pool. But in this case, we identify several epitopes which are present in the BA2, but not present in original Wuhan sequence. Then, for example, in this case, in the blue color, the Wuhan sequence, it is not working as a T-cell epitope. But in the case of BA2, the mutated in this the colored region, the red color changed from this blue color. But then in BA2, this sequence peptide, it can induce the T-cell response in this the BA2, the experienced the individual. So it means that this BA2 sequence, it work as the T-cell epitope, but Wuhan sequence does not. It means that the individual have this T-cell response after BA2 breakthrough infection. So we found such kind of the different example of the epitope, the two example more, the difference between Wuhan and BA2, and only BA2 sequence elicit the interferon gamma response in ICS by using PBMC who experienced the breakthrough infection. And also we found that, so now I'm presenting this table. So this is summary of this the three is the epitope. In one sequence, do not elicit this the ICS the response, so they are not T-cell epitope. But only this the BA2 mutated sequence, it can induce interferon gamma response in the experiment using PBMC from individual who experience the BA2 BTI. But then this is understandable. But then we found that these sequences are well maintained even in next strain, BA45, even in next strain, XBB, the 2023 last year sequence. And even we compare the current sequence, KP2 and KP3, among these two sequences are the 100% completely fully conserved. Then what's the meaning of this, the data? So this one is the very interesting part of our paper. So now we compare the CD8 T-cell response among three groups, the 3-dose vaccinated, and one more previous infection, 3-dose mRNA vaccine, and 3-dose vaccine and BA2 BTI group. Then, already I showed you before, the CD8 T-cell response against BA2 spike. This is the BA2 BTI experience individual, they have very boosted T-cell response by BA2 BTI. Then, how about BA45, this the next strain specific T-cell response. So when they got the BA2 breakthrough infection, BA45, it was not present in the world. So it emerged in the future. But then after, by using this the BA2 breakthrough infection experience, the individual, their PBMC CD8 T-cell, they have also boosted T-cell response against future strain. So this is really the expected considering this the sequence. So in this paper, so we could say that if someone experienced breakthrough infection, then they have boosted T-cell response against this the infecting strain, but not only infecting strain, but also future strain, so which are overlapping with this the infecting strain. In that way, so our immune system can be changed according to this the continuous infection or breakthrough infection and continuous emergence of variant. So this one is the one of the very interesting part of our research in terms of this the breakthrough infection and evolution. Just this is not exact term, but evolution of our immune system in the era of this the variant, continuous emergence of variant. Then, so let me move to my third topic. My third topic is also a very interesting one in the vaccinology. But now many people are talking about that. So during the COVID-19 pandemic, we got this vaccine by intramuscular vaccination. But then, so we need some mucosal antibody and mucosal tissue resident memory T cell for better immune response. So we might need the nasal vaccine or mucosal vaccine. Yeah, I agree with such kind of theory. But during the pandemic, I had some chance to look at what happened after vaccination in our nasal tissue. So I'm going to show you these data. So by the collaborating with the ENT doctor, ENT clinic, so we could obtain this nasal tissue during this sinus surgery. Then, so not only the peripheral blood, we also obtained the small piece of the nasal tissue, the mucosal tissue. And then, so in this tissue, also we have the three different groups. The first group is the unexposed group. It means that pre-pandemic, even these we started the collaboration not initially for COVID-19 pandemic. So we collected the nasal tip banking. So even before pandemic. So we had before pandemic 30 sample, unexposed sample. And also we have 60 sample who have vaccination, but without any infection. We confirm the absence of infection by assaying the N-specific antibody and N-specific T cells. The multiple stain, tetramer stain. And also, we obtained the nasal tissue from 26 the individual who have a vaccination and breakthrough infection. Then we use this AIM assay and MH class 1 tetramer staining in this population. At the beginning, without any this antigen specificity, we compared peripheral blood and nasal tissue T cell. In the CD69 and CD103 is well-known tissue resident memory T cell marker. So in the case of peripheral blood, the double positive cell or single positive cell nearly absent, very few. But then in the nasal tissue, 70% are double positive, real the TRM feature, and some not true TRM, but like tissue resident T cell, TRM-like T cell can be also defined by CD69 single positive, CD69 positive, but CD103 negative cell population was also like 14%. In that way, we confirm that really the nasal tissue T cell have the TRM feature. So regardless of SARS-CoV-2 antigen specificity. Also, we confirm again another the TRM marker, CD49A and CXCR6. All these markers are the highly expressed by this double positive or single positive. Then I'm showing you our tetramer staining data. So we successfully detected the MH class 1 tetramer positive CD8 T cell among nasal CD3 nasal T cell. But these are not only the breakthrough infection, the infected individual, but also vaccinated individual without BTI. And in terms of frequency of multiple positive cell, we found that between the vaccinated group and breakthrough infection group, the frequency are very comparable, not so different. And surprisingly, we found that the 6/9 and 103 double positivity. In this case, I can say that the double positive cell is the very real TRM cell. Their frequency was very comparable between these two groups. And C69 positive and 103 negative cell, we can say that TRM-like cell, they're also very comparable. Not only this marker, but also CD49A and CXCR6 the TRM marker are very comparable between the vaccinated group and breakthrough infection group. So with this data, even we can conclude that without any infection, only intramuscular vaccination, mRNA vaccination can elicit TRM cell in the nasal tissue. And then not only the frequency, but also we compare their function. So we stimulate them, the nasal T cell with the peptide T cell epitope. Then we perform the interferon gamma ICS, and then actually this one is the combined staining for tetramer and interferon gamma the ICS. And we found that interferon gamma production ability are comparable between the vaccinated group and breakthrough infection group. So we can say that even intramuscular mRNA vaccine induced the TRM cell exert very rapid effector function upon antigen stimulation with the similar degree to the breakthrough infection induced the nasal CD8 T cell. And finally, so we compared which TRM cell, regardless of the initial stimulation, regardless of vaccination or infection, which the TRM marker is good marker for the rapid effect function. And we found that CD90, the 49A, these positive cells, they have the much higher, the significantly higher frequency of interferon gamma producing cell upon in vitro stimulation. So we could define that among several the TRM marker, regardless of vaccination or infection, CD49A is an important marker for the rapid effector function. And also we confirmed that their TRM cell, tissue-resident memory T cell, so rapidly they show the degranulation function, like cytotoxic as a cytotoxic marker, and also they rapidly proliferate upon in vitro stimulation, peptide stimulation. So in this part, I showed that even mRNA vaccination, they induce the TRM, like the T cell in the nasal tissue, although it was injected to the IM, the route, not intranasal route. So this one is also one of the very important, I think the important finding for the future of vaccine development. And finally, I'm going to share with you the recent very unpublished data, but very interesting one. And what will be the effect of repeated immunization on memory T cell? So during the COVID-19 pandemic, many people have not only 2 dose, 3 dose, and 4th dose, in that way. So during the relatively short period, many people got the repeated dose. Then what's the impact of this repeated immunization on the T cell, memory T cell response? That was our question. So for this topic, the final topic, I'm bringing just a single slide, this one. And then, so if I make that very long term to short, so I can say that once we compare the second dose, and we compare second dose, third dose, and 4th dose, and we compare 6 months sample after second dose, and 6 months sample after third dose, 6 months after 4th dose. In that way, we compared after each injection the 6 months memory T cell response. And initially, although I'm not showing you the data, so we just stained them the tetramer, and we found that after third dose, it was the frequency of tetramer positive cell increased. After 4th dose, and further increased. So repeated dose increased the tetramer frequency. And also, we perform ICS, intracellular cytokine staining, and we found that the additional dose increased the cytokine production. So it seems that after 3rd dose and 4th dose, nothing to lose. It means that they are more enhanced, the more increased frequency and more increased cytokine production. So we can state that, more further vaccination enhanced our memory T cell response. But we found that, so we decided to look at the T cell response more in detail. So now I'm showing you one of the example. So now I'm showing our tetramer data and second dose after second dose, 6 months sample. So this one is direct ex vivo staining. So direct ex vivo staining, so we can detect this tetramer positive cell. And then we stimulate them, this CD8 T cell, PBMC, peripheral blood CD8 T cell, with the epitope peptide. Then we culture them for 6 days. Then after this 6 days proliferation, we stain them again with the tetramer. In that way, we could quantify their expansion capacity. During the in vitro stimulation, ex vivo stimulation, how many fold there's an increase of this expansion of this T cell population. We found that the second dose induced memory T cell, so in this assay, in average, they're like 20-fold expansion of this memory T cell. Then after 3rd dose, around 15-fold increase expansion. And after 4th dose, the 6 months memory T cell, they're 5-fold increase. So it means that this memory T cell, upon in vitro stimulation, they expand. But how about this expansion capacity? How much fold they expanded? So there's a gradual decrease after the repeated immunization. So we are now, this is an ongoing study. What's the mechanism? If there is some TSM cell is also involved, also we don't know yet. So now I'm performing some single-cell analysis to answer this question. But then, and also for the clinical question, why it happened. Just the repeated dose, it was a problem, or so very short period, every 6 months. They got every 6 months did the next dose. So this very short period is very important. We don't know yet. So anyway, this is ongoing study. So this is now our ongoing study by using the MH class 1 DNA barcoded MH class 1 tetramer. And now we are examining the mechanism why the different status is changed. So this is the almost end of my talk. So in my talk, so during the COVID-19 pandemic in my lab, we examined the SARS-CoV-2 specific T-cell response, mainly CD8 T-cell response, in terms of the TSM generation and their durability. And also what happened after breakthrough infection in terms of the epitope, the mutation, and also in the nasal tissue memory T-cell response, and also what's really the impact of the repeat immunization on the memory CD8 T-cell response. So so far, I shared our data from my lab, and so I have a double affiliation in KAIST in the university, and also the KVRI, the Institute for Basic Science. So from both sides, the KAIST and the KVRI, all this data was generated from my labs, and then especially by the So-Young Kim and Junyoung Go, Jae-Young Jung, and Sung-Min Jung, and Min-Ho Jeon, and Dr. Min-Kyung Jung and Sang Kim. Yeah, thank you for your attention. Thank you very much.
Thank you very much, Professor Chin. That was a fantastic talk, a really tour de force, and some really exciting data that was presented there. So I would like to open up the presentation for questions. So again, if participants would like to please either enter their comments in the chat function so I can see them, or else raise their hand or unmute. But maybe I will start off first and just have a question. So in particular, I was really interested with the comments that your latest data looking at the expansion capacity and showing a reduced expansion capacity of SARS-specific CD8 T cells with multiple boosting. And I was wondering, did you look at the breadth of response? So is the breadth of response also reduced if you look at multiple peptides at that, you reduced expansion, but what about the breadth of response?
So we didn't look at the breadth because actually in that study, ongoing study, we just focused on the tetramer in terms of our technique, the only tetramer, just 1 or 2. So only just in each individual, not whole breadth of epitope. We could use just only 1 or 2, O201 and 24 to HLA 24 tumor response. So we didn't look at so much the broad response. We didn't look at. So I cannot answer to your question. But that's a very important question, I think so.
And also further to that, looking at CD4 T cell responses, is there an interest in looking at CD4 responses in that capacity?
So yes, also again, yeah, I can answer to your question because in this ongoing study, we focus on CD8 T cell by using MH class on tetramer. So we don't know yet. But anyway, so by using this remaining sample, we can do that. Yeah, I will.
Good. Excellent. And just a follow-up question, also your really interesting data on the intramuscular immunization inducing CD8 T-cell, resident memory T cells in nasal fluids. So that was, to me, quite unexpected. And I was just wondering if you could speculate on the mechanism there.
Yeah. So actually, once I found that in the, not by our group, but I cannot remember exact the names of the investigator, but there are one mouse model data about that, almost same. But one difference, so they used the mouse model for mRNA vaccine, but in that vaccine study, they used the self-amplifying mRNA vaccine. But I think that the self-amplifying or non-amplifying vaccine is not the main matter for this study. But in the animal model, they also show that after mRNA vaccination, so tissue resident memory cell could be found in the nasal tissue, also other tissue also, not only in nasal tissue. But as a mechanism, we don't know yet. Even for another group, they could not answer about that. But one of my idea, the mRNA, a very small amount, very, very small amount of mRNA can go to the other tissue. It means that after IM injection, very small amount by bloodstream, they can go to the nasal tissue or other tissue. Then the spike protein can be produced at the site, tissue site. Very small. I did mind just the hypothesis. Maybe it is possible. So that's one of my idea. But yeah, so I don't know whether that's really the demonstrable or not. I don't know yet.
And so what would be your recommendation in terms of vaccination schedule, number of vaccines, the interval between them in terms of inducing robust, durable immunity?
Yeah. So for this, especially for COVID-19 vaccine, so I don't know yet. So one of my hypotheses, not just fourth or fifth vaccine. For example, flu vaccine. So we are getting flu vaccine every year, for example. So I think that there are two possibilities why this repeated vaccination reduced the expansion capacity in our study. Maybe just the repeated dose itself, maybe it's not the matter. But short period between each boosting for the next vaccine, it is maybe a problem because in our cohort, the third vaccine was given at 6 months after second vaccine, and fourth vaccine was given at 6 months after third vaccine. So in that way, so in next time, I'm trying to recruit some another cohort who have the 1 year interval, for example. Then it will be better, improved or not. I don't know yet also the answer yet. But anyway, in that way, I want to study different interval can result in different outcome. That will be very important for the scheduling. Yeah.
Yes. So I think, other studies have shown that increasing the interval is good for T-cell responses.
Yeah.
And what about the age profile of the subjects? Is there an influence of age?
Oh, sorry, I forgot.
Of age, how old were the subjects?
These are repeated immunization.
Sorry?
For which study? Repeated immunization?
In the repeated immunization, do you find a better response in younger people versus older?
So this repeated immunization, this cohort, is not the elderly people. Actually, this is also based on the healthcare worker. So our healthcare worker, their age, between 20 to 50, like mainly 30 or 40, more in that way. So not so elderly people. So also this kind of this repeated immunization, that impact maybe can be different between different age groups.
Okay. And what about comparing the response between, say, people that are 20 to 25 versus people that are 50 in terms of the breadth of response that you presented earlier in terms of like ability to respond to different variants?
So maybe you may remember that these are different age, have different TSM generation and different durability. Yeah, I saw it again. Also in that study, I didn't look at the breadth. So only the durability and the within tetramer-gating, their differentiation status. Maybe I can addition one more thing. They are not TSM cell, but other people, there's many publications about the T cell differentiation after this, like my study, and after vaccination. But in those people, in some case, the definition of this, the T cell differentiation, different subset, sometimes a little bit wrong. So we need I just want to emphasize that we need very cautious in the interpretation of this, the T cell differentiation status.
Good, thank you. Is there anyone else got any questions? I can't see any comments in the chat unless you can, Lars. Is there any more questions from anyone?
We have quite many questions in the chat, and I see also that Anke raised her hand, so maybe we can let Anke in to ask questions.
Right.
Oh yeah, now I can open the chat.
Yep. Okay. So Anke, do you want to ask your question, please?
Yes. Thanks a lot. Thanks a lot for a very interesting presentation. Actually, I have several questions, but I just do one now, and then others can also have their questions. So I was wondering, in the second part, you showed that after the breakthrough infection, there were only CD8s left specific for the new virus and for future viruses. But what actually happened to the CD8s against the original virus? Was there a loss of those? And why?
Oh, sure. maybe certain epitope which are present in the original virus will disappear after mutation. So actually not in our study, in someone else. Other study also they show that so the example of the epitope which abolishes, which disappear after mutation, after Omicron emergence. But we didn't focus on that. So maybe this T cell response will be the kind of balance overall, the losing by mutation and acquiring by mutation. But in our case, we focus on this, the acquiring, the newly elicited T cell response. Of course,
okay,
yeah.
I understand. Yeah. Okay, thank you.
Thank you. Okay, there's a question from an attendee. If TRMs are efficiently induced comparable to BTI, then there shouldn't be any need for nasal vaccination tried by many groups worldwide. So what is your opinion on that?
Sorry,
your opinion on the question is if TRMs are efficiently induced compared to BTI, then there shouldn't be any need for nasal vaccination.
Yeah. So yeah, simply we can say that. But so I want to focus that, emphasize that. I didn't emphasize that point in my talk. So even after breakthrough infection, so during the breakthrough infection, so really virus, it infects nasal tissue. But so I conclude that the TRM differentiation was comparable between vaccination only and BTI. But we need to think about that. CD69 positive, CD103 positive, double positive cell are regarded as a real, the true TRM cell. But this the real TRM cell, the percentage was like 10 to 20% even after the infection. It means that infection itself, in the case of the SARS-CoV-2, is not so strong, not so strongly induced TRM. So maybe, so by the development of better nasal vaccine, maybe we can increase the TRM frequency proportion among antigen-specific T cell. It means that in other terms, I can say that we can develop the superior nasal vaccine, even superior to natural infection in terms of TRM generation. So this is my answer.
Thank you. So there's another question about breakthrough question, and in particular about the study of what happens to the TSCM cells after breakthrough infection.
Oh, yeah. Oh, sure. So it's also the important one, but for this, you are asking of the newly de novo elicited one or old memory one. So I don't know yet. I don't know your question, but so I can divide into the two different type of memory T cell upon your question. It means that maybe old memory. Old memory means that so which are maintained from the original in the vaccination and then boosted by breakthrough infection. Such kind of memory cell, maybe also they will maintain some TSCM cell. But TSCM cell, sure, will be maintained. But then, so again, I can say another second type of memory T cell, which were newly elicited by this mutated sequence, de novo. In that case, TSM cell, initially the percentage was low, but gradually it increased, like the vaccination study. So they have two different kinetics.
Thank you. There's another question also very similar to the question I asked you about the expansion capacity, but in particular the question is what is the clinical significance of the reduced expansion capacity of CD8 T cells?
Oh, well, that's very, okay, yeah, very obvious because so when I examine the vaccine-induced memory T cell in vitro system, ex vivo system, so often I stimulate them with epitope or peptide epitope. So I can say that it is kind of simulation of in vivo exposure. Sure, this is the ex vivo in vitro. But then we can imagine that reduced expansion capacity in vitro, it means that in vivo situation, the host, they are exposed to the virus, antigen. Then what will happen in our lymph node or tissue? There may be reduced expansion capacity. So that's the kind of the significance of this, our in vitro analysis.
Very interesting. That is all the questions in the chat. Does anyone else online have any other questions if they'd like to? If so, raise your hand. No. So just, I guess, one last question that I might have is, again, in terms of the naive repertoire, in terms of like baseline cross-reactivity and how that feeds into the responses that you were detecting.
Yeah. So that's the reason. So to the Denise, the main question, I answered that I focused on the tetramer in terms of technique. So in my lab, we could detect this the SARS-CoV-2 specific T cell. So mainly 3 different techniques: the AIM, activating this marker, and ICS and tetramer. But in the case of the AIM and ICS, we could not exclude this baseline cross-reactive T cell. So when we use only tetramer, we could exclude this pre-existing cross-reactive T cell. That's the reason why in my study, I mainly rely on tetramer. So in the case of tetramer, I can say that there's no impact of cross-reactive, pre-existing cross-reactive T cell.
Yes. Yeah, the question was more about the other assays, so thank you very much. Listen, we are at time, so it was a real pleasure to hear your fantastic talk and your fantastic data. Thank you so much. It's a real privilege for ISB to have you join our webinar. So thank you very much, and thank you all the participants for attending, and Ted and Lars for helping behind the scenes. So again, thank you very much, and again, for all the attendees, I'd encourage you to look at the ISB website and please consider joining the Society if you are not a member. So thank you very much, everyone. Have a great night.
Thanks very much. Yeah. So can I go out now? Yeah.
Thank you.
Yeah, thanks very much. Yeah.
Thanks, Ted. Thanks, Lars.
Yeah.
Bye.
Thank you. Bye.
Bye-bye.
Bye.
Bye, Lars. Thank you. Thanks, Ted. Bye.